All right, some of you saw me already, and one of the things we decided to do was, put my mouth or my money where my mouth is, and actually present a life science diamond in the rough. It's one thing to talk about it abstractly, but it's yet another thing to actually, talk about one, and show why it's important. You already saw this graph. Okay, it was turned the other way in the other presentation, but it basically makes the point about how cancer is still an incredible, important scourge for us to be addressing. But one of the things that I think has been really interesting about cancer is over the last 40 years, cancer has been dealt with in a very blunt way. The observation was made early on that cancer cells grow fast. So the therapy must be, let's kill all fast-growing cells. And that's been the mainstay of chemotherapy and radiation as the main ways that we've treated patients with cancer of all types for decades. Only recently has the advent of personalized medicine and immuno-oncology started to change that paradigm. Well, one of the things missing from that is why are cells growing fast? What changed? I mean, they started out as normal cells. Why are they growing fast? What is the root cause of this? What is the mechanism? And that's what I'm going to tell you about. Because the Paslick story is a tale of what's actually causing cells to transition into cancer cells, and how can we take advantage of that mechanism to kill those cells selectively. So Paslick's has an entirely new mechanism explaining the cancer proliferation that's going on. Paslick's talks about, and I'll show you, how cancers are using this mechanism in a way that makes them vulnerable to a very particular kind of therapy. And we have an oral small molecule candidate already in development that allows us to selectively kill vulnerable cancer cells while leaving normal cells intact. And this works in vitro, but also in tumors, in mice, and the next step, of course, is to take that into people. So the key finding that led to this was the finding that a gene called NMT2 has a very low expression in tumors. Okay, well that's nice, it's a correlation of some kind. And furthermore, patients with the low expression of NMT2, they die faster. It's actually linked to mortality. If you just take this population of 470 patients with diffuse large B-cell lymphoma, and you divide them into two populations based on are you above average or below average in NMT2 expression, just the below average population is statistically significantly more likely to die. And it's because NMT2 acts as a tumor suppressor. And I can't decide which side I want to go to because I love pointing all this stuff out. So NMT2 is a gene that attaches a little tiny fatty acid to the end of a protein. And it does this for a couple hundred proteins along with its cognate NMT1. These two enzymes act in all human cells to help regulatory cells or regulatory proteins localize to the membranes where they interact with other proteins, because there's lots of proteins in the cell membranes. So this happens to be the only one of those two proteins that attaches that little fatty acid to the end of a gene called, or protein called naked1. And naked1 regulates a pathway called Wnt. Wnt pathway is implicated in all the cancer growth and metastasis. Wnt pathway turns on the expression of oncogenes. Wnt pathway turns on the expression of the genes responsible for metastasis. So by just attaching this little fatty acid to this one protein, this is suppressing cancer in cells all the time. It's a really simple process that's going on when you break it down. But what happens in cancer is that the expression of this protein has been epigenetically silenced. That's no longer turned on, that gene's no longer turned on, that protein's no longer expressed. When it's no longer expressed, you get no modification of naked1. It accumulates in the cytosol. When it's there, it does not regulate this pathway. This pathway turns on. You get overproduction of all these oncogenes, you get cell proliferation and metastasis. So one simple step allows normal cells to change into cancer cells just by silencing that one gene. So that's really cool. But it doesn't tell us how we're going to translate this into an actual therapy that targets cancer cells. So the real insight came from realizing that the cells that have this gene epigenetically silenced, they only have one copy of the enzymes necessary for controlling a couple hundred genes. And in fact, among those genes that these proteins control are the genes that control apoptosis. Apoptosis is the cell's process for programmed cell death. So it's also inhibiting apoptosis. At the same time, it's inhibiting cancer. So if you treat these cells with an inhibitor of that enzyme, the cancer cells are going to be a lot more sensitive to that because they only have one copy of this enzyme now than the normal cells, which have two. And the idea is if you give the right concentration of inhibitor, you're going to kill the cancer cells by letting them trigger their own programmed cell death through apoptosis and leave normal cells alone. All right, here's some data. This is showing some cells, cancer cell lines, normal cell lines. This axis is the concentration of the inhibitor that's being administered. So right here is the concentration that's killing the cancer cells. This is the concentration that kills the normal cells. It's about 100-fold different. So these are ultra-sensitive. And furthermore, the evidence is that what's happening in these cells, when you put higher concentrations of drug, these are all the markers of apoptosis that we're seeing. So it's actually turning on the mechanism that's predicted. So that's great. So one of the things about small company and academic discoveries is that they rarely translate into big pharma. And you get a drop-off of about 90% when you try to translate these kinds of academic discoveries into a pharmaceutical product. So this is a big pharma company running this drug against a panel of 131 different cancer cell lines, covering 6 different cancer types, both hematologic malignancies and solid tumors. 94% of the cell lines are sensitive to this drug. That's an incredibly broad spectrum of activity for a chemotherapeutic drug. And when you look at it in vivo and start looking at mouse xenografts, which is where you take some cancer tissue and put it in a mouse and let a tumor grow, when you test this against the drug, I'll do this over here for you guys this time. When you test against the drug, a small concentration inhibits tumor growth and a large concentration completely wipes out the tumors. And if you take that a little further and you actually look in the tumor at the activity of these enzymes, the inhibitor's doing what it should do. It's inhibiting the activity of the enzyme activity is going away, and that's correlated with clearing the tumors. And you can do it in human tumor cells too, so this is now taking it a step beyond the, we're going to do this in sort of cancer cell lines. We're doing it in tumors now. This is a human tumor, and this tumor has, this is normal cells stained brown because they've got the NMT2 expression. These don't have the brown staining. The xenograft looks exactly like the tumor out of the patient. And when you actually look at that xenograft, this is a almost identical plot. Intermediate or low level of drug stops the tumor from growing, high level wipes it out. And this is a drug-resistant tumor that resists all the other conventional chemotherapeutic regimens that are available now. So this is pretty exciting. Not only is there a completely new mechanism that explains the transition going on, but there's a drug that actually selectively inhibits and turns on apoptosis in those cancer cells. So anyway, what I was going to tell you next is I was going to tell you about the development plan, the team, and so on and so forth, and the markets for the opportunities. We're looking initially at hematologic malignancies because that's where we see the most activity, but we have looked at 19 different, we've looked at 19 different cancer types and looked at cells in cell lines in 19 different cancer types. We find evidence of this mechanism going on in some population of those cell lines in all 19 different types of cancer. And when we look at tumors, we see that the frequency that we see this mechanism in play so far, the frequency is 75 to 100% of the time. We've only covered 6 different types of cancers so far. Thank you. Okay. We've only covered 6 different types of cancers so far in the tumors, and we're surveying the rest. Because if this mechanism is really this ubiquitous, this is an amazing discovery in cancer. This is a real genuine breakthrough. Now, recently, a study was published that showed how a company was able to get approval for a drug in non-small cell lung cancer in 3 years from the time it first went into man until the time it was approved. So I know people are always talking about how long it takes to get drugs approved, how it's terrible, we don't want to invest, it takes forever to get our money back. This is first in man to approval in less than 3 years. So that's our target. We have a year to go if we could get in man, but as soon as we get in man, we want to be able to move just as quickly with adaptive clinical studies. So you've already met me, but I want to point out one of the individuals on the team whose research I've just shown you. Luc Betium is a professor at the University of Alberta in Canada, and he's an expert on this set of enzymes that does this activity of putting on that little fatty acid called mystrolylation. So there's IP. There's IP that's published. I even got an email today about another IP patent that was issued. So that's really exciting. Oncology is a great place if you want to do deals. So M&A is very hot in oncology. If you look at the last year of M&A deals here, which I'm summarizing, oncology was the hotspot for big pharma. So this is clearly an area where there are opportunities to make an exit. 4 of those top 10 firms have already been in contact with the company to talk about this compound and its potential. So this is a first-in-class oral small molecule. That's pretty exciting in and of itself. It's certainly different than most chemotherapeutics with a new mechanism, which is currently in review at a major journal. It's potentially effective against many different types of cancer. There is a companion diagnostic if we need it, but if it turns out that this is thus ubiquitous, the companion diagnostic won't make the cost-benefit analysis work like we talked about in one of the earlier presentations. So right now it's ready for pre-IND tox. We hope to be in man in a year. We've got a $1.5 million note we're floating because we can pull down an equivalent amount in Canada. And we'll follow that with a $25 million Series A later this year. Thanks a lot. You can certainly contact me about this company or anything else. And this is why it's a life science diamond in the rough. Okay? You can't get farther out of the mainstream than Edmonton, Alberta. Okay? Anyway, thanks a lot for listening.